Next up, we have the panel discussion. Last year, when we had the conference here in Cambridge, it's definitely easier to have a panel discussion, in a room with everyone there, where we can make it very interactive. It's going to be harder doing it digitally. We're going to give it a go. We'll try our best. The theme of the discussion is around the importance of understanding the biological origins of VOCs. We've got a few seed questions to get the discussion started, but what would be great as we go through, if you have additional questions or want to clarify specific points or put specific questions to individual panel members, do that through the Q&A type function. Equally, if you have a strong view on it, fortunately, we can't unmute everyone's microphones. So we'll need to kind of rely on the typed and written responses for that. We've got the panel, I think, pretty much all here. So if they could unmute microphones and share video. We'll also have some of the earlier panelists as well, who can chip in with any perspectives and answers that they have. So what I'd love to do is just get the individual members to quickly introduce themselves. So we've heard from most of them already through the conference, but just a quick who you are and one or two sentences just on your background to refresh everyone and to introduce for those who may not have been part of the discussions earlier today or yesterday. Let's start with you, please, Mark. You're on mute.
Yeah, sorry for that. So, hello, I'm Pierrick Stefanuto. I'm from the University of Liège, and I'm a senior scientist, and I'm analytical chemist first, so I'm mostly working on the technological side of VOC and all type of volatile-omics research.
Thank you. Mark, could you just give a quick introduction?
Hi, Mark van der Schee, medical doctor and a biomedical scientist by training, been involved with Alston for about 5 years now.
Fantastic. Terence, could you just give a quick introduction?
Terence Risby. I am a retired professor from Hopkins. I've been playing in breath research since the 80s. I started just because the problems with infection of blood and urine with HIV, and now we've come the complete circle. Now we have breath that is also infectious.
Thank you. And Rianne, can you just give a quick introduction?
Hi everyone, I'm Rianne Feiten. I'm a senior scientist at a cancer therapy center in Maastricht in the Netherlands. I did my PhD in breath omics. I actually don't really do it right now. I mainly focus on data science in the clinic at the moment, but still very interested in breath omics, of course.
Well, thanks so much. As I said, please do submit questions as we go through. I'll kind of kick it off with a few questions that we have here. So, one of the things that kind of strikes me is when you look in standard metabolomics, you're kind of building off a lot of well-established knowledge in biochemistry. So kind of first question is, where do you think we are now? How well do we currently understand the biological origins of VOCs? I'll kind of go through everyone individually, and then we'll open it up for a bit of a broader discussion. So Terence, maybe we could start with you, if that's okay.
I thought that was coming. I think there are a number of compounds that we understand their origins. We understand the origin of molecules like isoprene, which is in the biochemical pathway of cholesterol. We understand the production of ammonia and amines because they're in protein metabolism. We understand oxidative stress and the products of aldehydes and hydrocarbons. We understand products like dimethyl sulfide. This is a production of tissue death, which is quite an involvement in the liver. So I think we know a number of molecules. Personally, I have no understanding where fluorinated compounds are coming in breath. I think they're environment. I think there's a problem with a lot of things where we're measuring, well, breath analysis, breath contains molecules from metabolism, from exposure, what you're exposed through the skin and through and inhaled, and then finally at very low concentrations, they're involved in changing in metabolomics as a result of disease or normal processes. My view. Okay.
Do you think that I think there's obviously some specific challenges with breath as well. Do you think that's also the case in blood-based metabolomics, for instance, where you have quite a broad range of exogenous sources as well? So do you think that's
absolutely clear? There's getting the molecules out of the cell into the bloodstream and from the bloodstream to the lung is an important parameter. I think we heard today in the discussion about dialysis that plays a role in the excretion. They're changing as a result of going through the dialysis interface. You're going to change the concentration as a result of dialysis. So I think that's an important part of breath per se.
Thank you. Pierre, do you want to kind of take that first question? Where do you think we are with biological understanding today?
Yeah, for me, I really relate to what Terence just said. It's the fact that I think we have a good understanding of the biology and how the compounds can be produced by the body, but we still don't know how they react along the way from where they are produced to the machine. And of course, there is all the change that can happen in the body. They can be like used in another cycle from another organ. They can be like trapped somewhere like in cells or in the lung, but also after like how we transfer that into the machine. So really like the sampling device that we will use, like we try to be untargeted, but we know that it's never like really truly untargeted. So I think that when we say that we have a breath marker or a good candidate, it's one compound that is linked to the phenomena that we measure and that a bit by chance make it through the whole process until we can detect it with our model at the end.
Excellent. Thanks, Fabian. Anything additional to add there? I remember from your talk a couple of years ago, and you obviously kindly helped write a blog post as well, where you were relating your breath right down to what you're saying, other types of modalities as well.
Yeah, so I think I'm a bit more skeptical than Pierre and Terence actually. I actually think, like, for example, lipid peroxidation, right? We say, okay, we know which compounds come from lipid peroxidation, but why then do we measure some of these compounds in one disease and some of these compounds in another disease, right? We also don't know where they come from specifically in a pathway. They're not in the pathways usually. And this is already an issue with metabolomics where, you have a lot of genetic pathways and some metabolites are now in those pathways, but that is not the case for the volatiles. So I actually think there's still a lot that we don't know. And I think if we want to actually link a VOC to a disease or to a process that is altered, then we need to look a lot more in depth than just very broad processes like lipid peroxidation and oxidative stress.
And do you think that's a tractable problem? So if we have better ways to capture and analyze, can we relate it back to where those types of VOCs fit within specific parts of the pathways?
I think it's difficult. I think you have to start with finding associations, right, between a disease or a process and VOCs. You could do that in vitro or in animal studies. And in the end, what you need to do is if you have an association, you need to prove that it's a causal relationship, right? And then you need to think about things like, I don't know, knockout mice or even cells where you knock out a process and see what happens to these VOCs. That is the only way you can actually establish a causal relationship. And I think we don't really have that yet. I mean, for stuff like isoprene, we know that, right? But I think a lot of the other stuff, we still don't have any idea, except for that they're sort of associated with these larger processes like oxidative stress. But oxidative stress is also a multifaceted process, right? It's an interaction between different pathways. So I always still find it a bit too broad, I guess, if you know what I mean.
Yeah, no, it's interesting. Well, Mark, do you want to have a go at this one? And then what we'll do is kind of reflect back with the rest of the
I think sort of conceptually, I'm more in the camp of Rihanna, almost, where I think we're just starting to scratch the surface, and it's actually quite important to think about what the position is of sort of biological validation within your experiments and how you can incorporate that. I think understanding the biology of the targets you find is actually quite critical. I think PH has shown some very interesting research in that direction through their in vitro models. And I think what we'll see in the community, that more and more people will develop these kind of models that allow us to probe that biology. In that context, I'm quite excited about sort of two streams of work. One is sort of the increasing emphasis on fluxomics, where people are interested to understand how, if you provide a certain a labeled compound, how it flows through the human biology and results in a specific product. And related to that, I think the whole EVOC probe approach is one that is very compelling, because it allows you to utilize a lot of the existing knowledge of biology. And it's fair to say that existing genomics and proteomics have a higher understanding of the underpinning biology than breath omics. So you can actually sort of utilize that knowledge and try and bridge the gap. But bridging the gap, in my mind, is still definitely needed.
Excellent, thanks for that. So maybe looping it back to Terence again, Pierre, if there's any additional kind of thoughts you wanted to touch on or hearing from what the others have said. I think you're on mute, Terence.
I think oxidative stress is so generic that, it is depending on where the tissue is, what fatty acids are in that tissue. I think it's very generic. And I think that's teleologically, that's normal development of of aerobic systems and anaerobic systems. So I don't think, I think you can measure oxidative stress, but I'm not sure that it's ever going to be disease related. It's going to be host response. So that's my view. So I don't disagree with what's being said. I don't think anybody should claim that oxidative stress is a diagnostic test. I think it's designed to be host response, whether it's an infection or disease. The body has to defend itself, and that's what oxidative stress is part of. That's my view.
Excellent, Pierre. Any additional thoughts there?
Yeah, like for me, like as it was mentioned by Mark and Ryan, I really think that, yeah, maybe we can go deeper in the understanding of the chemistry and the specific process, but there are like so many things taking place at the same time that I wonder, like, if we can really like get really specific, and basically if it was the investment to be that specific, if we can like just generate something that allows us to understand, okay, this is more like an inflammation type of response, or more like a infection-based response, and just like model that will be already like a huge development, just understanding like general processes than like probing. Like I would dream, like as Mark mentioned, we are trying to do an asthma in vitro model. I would dream to have a C13 label, like compounds that we can degrade and see really like where, for example, non-anal is coming from. But there are so many sources of it that, like, I don't know, like the size of the study and the type of control that you have on the genetic material of the model to be able to do that.
Interesting. That kind of actually leads into the second question, which you've touched on a little bit there. When you think about the just trying to analyze what may be present versus administering substrates or probes. Like when you look at some of the breath tests which are commonly used today, H. pylori, Anthony yesterday when he was talking about some of the gut tests, the hydrogen methane testing. You're administering specific probes and substrates, and that's obviously a lot of the work that we're focused on with our VOC probes as well. So it's kind of, do you think we should as a community push harder on that? Understand a particular underpinning piece of biology and trying to hit it with a specific probe and get way above the baseline, introduce unique compounds, or do you think continuing to do discovery based work where you're trying to capture as many compounds as possible, analyze those and relate it back to different questions that you're trying to ask? So it'd be interesting your kind of perspective on the balance of those and if there's a kind of one tipping in favor of the other. So maybe we'll go through the same order this time round again, and we'll mix it up for the next question. So Terence, maybe start with you again there.
I honestly think that the cleverest research in breath is know the biochemical pathway and give a probe molecule and with stable isotope or what have you. I think that's a very powerful approach. It is very useful, but let's be practical. That's a very, very expensive research to conduct in terms of getting approval for the substrates that you're going to get them approved through in the US, to get FDA approval, and then to approve not only the derivative, but also its stable isotope. That's expensive research. And also, it's not really that sexy. I mean, let's be honest, since COVID-19 has been around, everybody is trying to find a marker for COVID-19 because that's sexy. So I think, there's things you can do as an individual researcher. That's discovery. Trying to develop, clinical tests using stable isotopes, that's expensive science.
Interesting. Thanks for that, Pierre. Do you want to pick it up from there as well? I think obviously stable isotopes is one approach, but one of the things that strikes me about the gut health ones is actually those substrates are incredibly low cost because it's glucose and lactulose as well. So I think whether or not you can emulate that paradigm and pick safe, cheap substrates is another approach that you can think of. But maybe Pierre, do you want to pick that up from there?
Yeah, I agree with the cost of isotopes. So that's expensive research, but I think also it will be necessary at one point if we have really a specific target that we want to hit. So I imagine that I'm working a lot with inflammation, and we have some stuff that are like related to inflammation. So maybe we can have like a molecule that is really like specific to reaction with reactive oxygen species, and we can try to like probe this mechanism and say, okay, in this patient we have a lot of this species and we can see a signature. And I think that we have to first study that with isotope label, and maybe, as you mentioned, having like a cheap substrate will be like the way to go probably. But I still think that if we want for one specific thing, like monitor and understand completely the chemistry, we have to do it like through like expensive science. And I agree with Terence, like it's not sexy, but I always found that proof of concept is sexy. Oh yeah, I have this like breast tool, I can do like a really fancy research with that. But when you say, okay, you have to do like different phase, like blind study, like no one is doing that because this is the boring part, but it's the necessary part to make breast like a really like useful tool in the clinic.
Yeah, no, thanks. I agree. Yeah, boring is good if it gets you to the point where it works. Exactly. Brianne, do you want to pick it up from there?
Yeah, I think I don't think they are mutually exclusive. That's the main point I want to make. I think we can do both in parallel, because I think the biological origin question that we're talking about here is not the only problem that we have with breathomics. One of the things that we all know as well, I saw someone mention about brushing teeth before, getting your breath analyzed. The variation that you see between samples is also still a really big problem, right? Depends on what you've eaten, depends on whether you had deodorant or not, right? So I think actually the more clinical studies that we're still doing with the non-targeted mass spectrometry, for example, are still very useful, and we still need to figure out how to reduce this variation, or at least how to deal with it. So I would say both of these questions are really important, and we should do both in parallel, I would say, whether they're sexy or not.
Thanks for that, Mark.
Yeah, I think I mostly agree with what the previous speakers have said here. So I fundamentally think it's going to take a long time, but if we get to this point where we can build this map with our understanding of the VOC biology, it will make all the research we do going forward that much stronger. If you think about a genetic experiment, you go online, you check the human genome, you know what the relevance is of all the genetic readouts that you find. Obviously, that's much more challenging with a fluctuating metabolomic sample, but I'm still hopeful that we can get there in the future. And I'd almost say, in my mind, the probing and the fluxomics started with the C13 labels, but in effect, even that brushing of the teeth that has been mentioned can be seen as a probe. If that changes your breath signal, the speed at which it washes out, maybe it's just breathing kinetics, but if it's metabolized in any way through the body, it's effectively acting as a probe in its own right. And I think the interesting thing there is with those kind of probes is the readout can be multiplexed. You're not just looking at a carbon label 13, you could actually look at the wider space of molecules and start to combine multiple in order to probe more diverse ranges of biology. So I think it will be a challenge, but I'm reasonably optimistic we'll get there as long as we combine efforts in that space.
Yeah, I think that's an interesting point. Like, so obviously in the session yesterday, the liver session, limiting was discussed quite extensively there, and that obviously comes in through ad hoc data intake as well. And I think what was interesting about that, when I first saw that, in my mind, I was always like, okay, well, exogenous source, it can't ever be useful. But I think there that shows the potential to look slightly broader and see how those things that we may have, chucked into the dustbin of chemical noise, if you like, or exogenous sources, may be repurposed for something that could be diagnostically quite useful. So one point that's came through from Neil, just related to stable isotopes, says the panel is correct in saying development breath test of stable isotope is expensive, does involve PMA application, however, it will work. So one of the things that was touched on earlier, it relates back to the design of some of the experiments. So to what extent should we use in vitro and ex vivo data for identifying and confirming the origins of VOCs in breath research? Because often I hear conflicting views on this. So you may do something in vitro, but you've still got chemical noise issues there and so right. Is it translatable? Versus the counter argument, if you go into human too early, it's a very complex system. You can't isolate what it is that you're looking for. So would be interested to hear the range of views on that. Maybe we'll mix up the order a little bit and start with you, Brianne.
I think in vitro systems are the way to go. Simply because if you look at the other omics fields that are ahead of the breath omics field, I mean, it's as simple as that. Even like genomics or transcriptomics, the only way you're going to figure out these associations between gene expression, metabolites, and VOCs is by looking very much in detail into the molecular pathways. And the only way you can do that in a controlled setting is cells. So I think we should start at in vitro. And the same as with all the other omics fields, you then progress to animal studies where you look at a slightly larger scale, and then you go to humans and try it there, or at least investigate there. So I think that is the way to go, because otherwise we're just in the dark without a flashlight. We have no idea what to look for, right? And we can't see anything. So we have to start somewhere, I think.
Yeah, do you want to pick it up from there?
I can agree on one part of what Rian said, that the fact that in vitro is really important, and it's really like, oh, we will like make sure that we understand what's happening. If we have really important compounds, like how can we generate it in vitro, will help us to understand where it's coming from and what's the variability that we can expect. Using that as a like building up approach, like going from cell to like animal model to human, I think it's really difficult. And for now, it's mostly on the technology point of view, because I think that it's really difficult to trap VOC the exact same way in a cell box that is growing in a mice or like in a human. So I think that just because of the analytical technology after the biological process, it's really difficult to make it translatable. It's what we were discussing before about like the origin of the compounds. Like in a cell, maybe we will generate the exact mechanism that we are interested in, like oxidative stress or whatever, and we'll have these compounds. But in a human or in a mice, it will be like metabolized on a different way before it arrives to the lung and is transferred to the breast. So yes, it helps to understand the science. I think it's important for the robustness, but I'm not sure about like translation from one to the other. Like maybe like, or maybe it would be by group of molecules, like saying like, oh, I see an increase on aldehyde. I saw a lot of small one in vitro, but the bigger one are in vivo because they travel better through the lung. I don't know.
Mark, do you want to pick it up next?
Yeah, I share that viewpoint that I think for discovery, it's very challenging. The systems used for discovery in vitro settings, I don't think they're necessarily proven to be less noisy than doing in vivo analysis per se. So it generates loads of challenges around translation. I think for proving a specific molecular mechanism of a target volatile, again, I think it would be quite useful there. And given your background, Rihanna, I would almost sort of just throw it out there. With your viewpoint as a data scientist, wouldn't you say, let's start, for instance, by profiling a large portion of the healthy population, and then saying, okay, now we have our reference values, and if we do a study in a cohort, and we see that there's deviation from that baseline, that would make it an interesting target for us and could help us to deal with a lot of this variation and natural complexity that occurs that is not disease related.
I think that could work. And then you need a lot of samples, obviously. It's like the epidemiological studies that you need thousands of samples. I mean, it's doable, but it sounds expensive. We could definitely do it though.
Excellent, thank you. Terence, do you want to chip in where your perspectives are on, and I suppose animal models kind of fit into this as well when we think about in vitro and ex vivo?
I think that everybody's saying important things. We did a study where we were looking at with genetically mice, knockout mice, and looking at obesity, and found that one of the markers was ethanol. It's in the gut, and you could change the gut metabolome and change the gut, and you could decrease things like NAFL or NASH, non-alcoholic steatohepatitis. But that was a great study, but we're dead now because everybody's using alcohol. So you cannot go to a clinic anywhere in any hospital in the world where there's not ethanol, high concentration of ethanol. So there's advantages to go and start with an animal model and then go into the clinic, but you have a problem there with what's going on in the clinic, and those studies are dead. We had approved studies to go on obesity and non-alcoholic steatohepatitis, and they're dead. So you have to be careful. That's my call. But I do believe that start with the animals and go to the humans.
Interesting, thanks for that. So we've had a question come in. So obviously we touched on in vitro there. When we think about VOCs, obviously we've been focused on breath as a sample metrics, but there's a question posed to what extent are other sample matrices that have VOCs, saliva, sweat, urine, etc., how important a role is that when we try to kind of dig down and identify those VOCs that do have true biological origin. So if you have views on where and how other sample types play a role in here, I think that would be fantastic. So maybe why don't we start Pierre on that one first?
Yeah, I think like other type of sample is really interesting, like because like in the idea that we were speaking about, like pathway discovery and everything, like taking like different like layers of I would say like volatility or size of molecule in different like matrices, like starting for like breath, like really the most volatile, after maybe some EBC, some saliva, some like brushing from the lung, we could like really see, like for example, we were speaking about oxidative stress, lipid peroxidation. So it's interesting to have a liquid matrices where we can do lipidomics and see how the lipid profile is changing and how it's affecting the VOC profile. So this is like really like in terms of translation and understanding a really like great thing to do. And I think now with the development of like the multi-omics like processing capabilities, I think it's really something that will happen like soon in the field.
Thanks for that. Terence, you want to take it up from there?
I'm not sure I can add something right now to the discussion, to be honest.
Yeah, that's no problem. Mark?
Sorry, sticky mute button. Yeah, so I think it's an interesting one. In my mind, it's almost not the sample that matters or doing VOC analysis in other samples. I think what can be interesting about other sample types is when you can perform multiple analyses on the same matrix. And sorry, that's a bit abstract, but for instance, when you think about, I don't know, well, breath in a way is also a good example. If you would have the exhaled aerosols and you have the volatiles, because they originate from the same organ, the same area in the body, it's actually quite interesting to see how these tie together. There could actually be, because volatiles will have differential solubility, so it may be actually that the cumulative value between what you find in exhaled aerosols and in breath could add information. There may be some processes you can only learn about in the exhaled breath droplets or aerosols that result in production of VOCs. And I think that may be similar for other matrices as well, where you do see, for instance, in blood analysis, where people have one sample, do genetic, proteomics, transcriptomics. That's when you can start to look at this multi-omics that Pierre Hughes mentioned. And perhaps that's a direction where breath can move into as well, with combining EBA and VOCs. I haven't seen anyone do it yet, but that would be quite interesting.
Rhian, anything to add on that question?
Yeah, I wanted to add one small thing, I guess. I actually think, especially since there is a lot of variation in breath sampling, and breath analysis between people and even within a person. I actually think, if we want to make it clinically applicable or useful, combining it with another source of information is actually the way to go. For example, blood, like Mark said, or maybe urine, I don't know. Combining those different sources of information about a disease will really help, I think, in increasing the accuracy and reducing the variation. And then for the multi-omics, just wanted to mention that I think that is actually the way to go for in vitro studies, right? Measure gene expression, measure metabolites, measure VOCs, and try to connect them together. That's a side note.
Yeah, no, that's very interesting. And the kind of question relating to different sources of VOCs, it kind of reminds me a little bit of some of George Hanna's work done in Imperial, where in one of his studies, he was looking not just at breath sample, but at resected tissue, so ex-vivo sampling, and also endoluminal sampling as well. So getting down in there, so different ways of sampling and analysis, and seeing where you have a degree of overlap with some of the reported VOCs. So I think attacking it from different directions does make a lot of sense. So another question we have here is related to VOCs and the microbiome. So to what extent, how relevant are VOCs to the microbiome, and do you view these as exogenous or endogenous, and what the potential applications may be there? So maybe we'll start with Terence on this one.
I honestly think the microbiome is incredibly important in its contributions to breath. Absolutely. And it dominates in many ways. And controlling that is going to be diet. And this is a big, huge variable in all breath analysis, the microbiome. And that is why, people take breath samples when people haven't eaten for 12 hours, but that sort of limits you to not using diabetics because you're putting them at risk. So I think it's important. I think it's absolute. And you can manipulate the biome, giving, probiotics, giving, asking a subject to eat yogurt, et cetera, et cetera. You can do all these things, but that's a big variable that is around. I mean, there's also a big variable on the way a person breathes. And we've seen it. So adding that, that's a confounder.
Yeah, I totally agree with that. Pierre, do you want to pick it up?
Yeah, I think just to reference to Terence's discussion yesterday, basically to rule out microbiome variation, we have to use one patient as his own control. This is the way to go, because all what Terence just described, it means that you need an individual that is basically your clone. So it means you have to be your own control, and this is how we will minimize variation. If not, I think we will, with all the growth of microbiome, we'll have more and more information about that. But to really understand it and comprehend the impact on breath, it will be really difficult, because microbiome signature is our DNA. It's highly specific. So we'll never manage to completely control it. We just have to know it's there, and this is part of the background variation that we see in breast study.
Brianne, do you want to pick it up?
I don't have much to add. I agree. I don't think it's feasible to control for microbiome. I do think it's really important because it's sort of a symbiosis, right, between the human body and the microbiome. But I don't think we can do much about it, to be honest.
Can we use that to our advantage in the sense when you think about the clinical applications? So obviously a lot of the discussion that Anthony focused on yesterday related to gut health, kind of targeting the microbiome with different substrates.
I guess depends on the disease, I guess, right? For some diseases it might be relevant, for others it might not. But yeah, without further testing, you're not going to find out what kind of microbiome a patient has, right? So whether it actually has any benefit for diagnosing a disease, let's say, I would find that not really feasible, I guess. Because then you're going to do extra tests, and you might as well just do the golden standard that they use now, right? Because then breath almost becomes more complicated even than that. So yeah, I'm not sure, I guess.
Okay. Mark?
Yeah, for me, almost a microbiome question is one that relates to what we discussed just previously. It may be actually a very interesting orthogonal piece of information. In the microbiome field, people are struggling with the fact that the presence of these pieces of DNA doesn't tell you much about the activity of these pieces, and that varies widely. In VOC research, we struggle with, okay, there's these VOCs, but what microbes do they actually come from? So the way we've been starting to think about it is actually that there could be a lot of complementary value there. And I showed a snippet of data during the COVID talk today where we're exploring if you can use one sample to collect VOCs and also your exhaled microbiome. And it's that kind of stacking of information, which I think especially in chronic inflammatory and infectious disease may be very relevant going forward.
Excellent, excellent. So a quick reminder, if you do have any questions, post those in the Q&A box. One question which is a little bit, I suppose it relates back to James's talk earlier. So several VOCs have signaling roles in biology. Do you believe there could be others? And then the follow-on question is, do you think VOCs could actually, if that's the case, do you think VOCs could be deployed for potentially therapeutic uses? Let's start with Mark on that one. Mix it up a bit.
Sorry, what was that last bit?
If VOCs have signaling roles, ultimately, could you think about the use of VOCs as a therapeutic?
Yeah, I think that's something that's been really fascinating me actually over the past few months. I think we've always looked at VOCs as passive molecules, but actually, if you look at the evidence with aldehydes, particularly, they seem to be part of sustaining a cycle of oxidative stress. So perhaps intervening in that cycle, you could actually envision that you are able to then break that positive feedback loop. And there's, for instance, an example where exogenous oxide is used in neonates with bronchial pulmonary dysplasia. And obviously, in some way, anesthetics are inhaled anesthetics are a bit of a volatile therapeutic as well. So yeah, I think we're far away from it. It's sort of blue sky stuff for the future, but I don't see a reason why we couldn't end up there in the future.
Okay, Rhian, do you want to pick it up there if you have anything to add?
So my knowledge of VOCs is not really up to date. I Googled a little bit today, but what I remember from when I finished my PhD is that, at least as far as I could tell, most of them are byproducts of processes, right? But like Mark said, it seems that some of them might actually be part of processes. But I would assume that most of them will still remain byproducts. And so then I guess if you want to use therapeutics, then you should really look at the process that they are derived from, obviously, to find a therapeutic target. Not sure if the VOCs themselves will be targetable, at least not many of them.
Yeah, anything to add there?
Yeah, nothing really. I think we all agree that it's like something that can have a big impact, but we have no enough information. I think to all the also traditional medicine where people are inhaling like some like essential oil or like any type of molecule that can have like anti-inflammatory effect, that maybe it's also a way to go, and maybe we have some interaction on the volatile level, but yeah, this is just hypothesis.
Terence, anything to add there? I saw you nodding when Mark mentioned nitric oxide as a treatment.
I agree with Mark. Nitric oxide, carbon monoxide are definitely involved in signaling. I believe that isoprene is involved in signaling. There probably are other molecules that are involved in signaling, but I think these are molecules that are somewhat reactive, so that they can be turned on and turned off. And aldehydes are good examples of potential markers, because clearly they can be oxidized very easily. So that's I doubt hydrocarbons are a marker of signaling, because hydrocarbons are pretty labile. So maybe ammonia is a marker. Ammonia is incredibly difficult to measure in breath because it sticks to everything. Many of the studies, in terms of patients with cirrhosis, the treatment is essentially to give people lactulose and give them violent diarrhea as a way of getting rid of the ammonia. So I think ammonia is important. I don't know how important it is, but I think the molecules are reactive.
Very interesting. I did not know that. One question has come in here. So would the study of metabolic disorders like inborn errors of metabolism help more in understanding specific VOC pathways in vivo? Why don't we go in reverse order there? Terence, maybe you want to start us off on that one.
We did some studies in neonates, and we were looking at neonates that are oxidatively stressed, and we looked at the efficacy of vitamin A, vitamin E. That's about my own experience in looking at that in neonates. I don't think people have done many studies in this area. I think it's definitely possible to do this research. One of the things is actually studying neonates. Nurses in neonatal intensive care units are were very protective of their children, and they don't really like us collecting breath. I know that from studies that we did in the past. But yes, this is the way to go, and this is where you can do it with knockout mice.
Thanks, Pierre. Anything to add there?
Not really. I have no much experience, but I think like all what is described is really interesting there in that field.
Ryan or Mark, stick your hand up if you've got additional points to add on that question. Mark?
Yeah, I'd see this, I think, to the point of that was made earlier. It's almost a human knockout model. So I'd almost see, because a lot of these diseases are very, very niche in a way, unfortunately quite rare, and typically there are perfectly fine diagnostics out there for these diseases. So I wouldn't necessarily say, oh, this is where breath tests can be most impactful, but it could be a very interesting model to say, hey, I think this is related to this and this metabolic pathway in the liver. Let's try and find a disease and a small group of patients that have a complete knockout of this pathway and use them as a human knockout model. And yeah, I think that could be quite elegant if you want to study flux.
Excellent, thanks, Chad. So one comment has come in just relating back to the signaling from Rob. Microbial VOCs, e.g. terpenes, have a signaling role. Plants also utilize VOCs as signaling molecules, defense warnings, etc. So one of the points that's been raised a few times over the last couple of days relates back to the confidence in identifying the actual compound. So obviously, if we are to get a better biological understanding, you need to know what the thing is, and that's hard enough to do even when using things like mass spec. And we still kind of see a range of different analytical techniques being used in the breath space that kind of fall into the spectrometry based like mass spec versus more pattern based eNOs, etc. So I wanted to get your opinion on how best to use these different tools, and at what point you should start to think about using these different tools within breath research. So maybe we can start, Pierre, with you on that one.
Yeah, like I think like compounds identity validation is really like the core of the future of the field. Like when we were speaking about like boring part, like no one wants to like validate identification, but without that we continue to publish like list of compounds that we don't even know what they are. And like for me, like something that we have been discussing for a long time in the group is like we are a GCMS group, and we are using like mostly impact ionization, which is really bad for hydrocarbons. And we see like so much hydrocarbons in breath that we are like, maybe we have it wrong for the beginning, or we say that we try to find markers that are not hydrocarbons, or we change the technique that we are using to be able to identify those hydrocarbons compounds. So I think this is like really interesting to see like everyone mentioning that and the need of validation, because I think it's really what it's lacking now. And like, after the way to go, there is plenty of guidelines in metabolomics in general that we can apply to VOC. Like we discussed like this morning about like the metabolomic standard initiative that you can like really translate that to breast and like get some like standard for validation, because, and again, for that, hydrocarbon is a nightmare, because having a branch hydrocarbon pure standard, like it's hard to find.
Ryan, anything you want to touch on there in terms of the use of different techniques and challenges?
Yeah, I agree with Pierre about the identification barrier, I guess. It's a big one, right? But, you know, if we look at the electronic noses compared to the MS based methods, I do still think, I've actually never used the e-nose myself, but I do still think those are most likely to be successful in the clinic, because they can give you like, you know, an answer quickly. They can do it within the consultation room, let's say. I think that would appeal to doctors a lot, instead of sending samples away like they do with blood work. That's what I've seen anyway in the clinic where I work now. They're interested in low threshold things that give results quickly. With the VOCs, I always see them as, or the MS, I would like to know why things happen, right? So I guess the MS would be the first step. If I had to do it, I would first start with MS to find out which VOCs, you know, are found for a disease, and then move to an e-nose that we can actually use in the clinic. That would be the ideal approach, in my opinion.
That's interesting. We'll move on to Mark. I suppose we're like, one of the other points is, you know, you don't quite have an equivalent of e-nose for blood-based analysis. So it seems that pretty much everything is done within the lab. So it does seem that that is, from a workflow perspective, reasonably accepted. But maybe Mark, do you want to kind of?
Yeah, I would actually.
Well,
I would echo that from at least my personal clinical experience. Clinicians are lazy and conservative. I feel comfortable saying that, at least still being an MD myself. But if it fits within the current paradigm, and it's actually a nurse that takes the sample and it flows through this pipeline and you get results for many tests, that's absolutely fine. There are specific tests like, okay, patients at the ER and what's happening, is this a viral infection or bacterial infection? That's where an immediate test could have an enormous amount of value. And I fundamentally agree with you on the principle of, in those cases, that a point of care approach would be extremely valuable, and it would effectively be the only option. I do feel the way to get there is like you're describing. First, understand what you're trying to detect, and then make sure that whatever sensor you pick is the most sensitive one to do so. I know I come from that background originally, and during my PhD did mostly e-nose work. But one thing I really took from that learning was that because you don't know what you're measuring, actually I started seeing drift over time. And is that relevant to my classification or not? And how do I calibrate my device? Because I can never calibrate it. If you would know what the compounds are, you can pick the best sensor and you can calibrate it with a mixture each time, because you know it needs to detect X, Y, and Z. So I definitely think they have value, but I'd almost say we're not at that stage yet. And that is also translated in the fact that the only successfully used sensors in the breath context in the clinic are the C13 ones at this stage. So, and exogenous nitric oxide, of course, but that's also a known compound, so very focused.
And hydrogen methane.
Yes. But again, known compounds.
Terence, do you have any kind of thoughts on that kind of?
I agree totally with Mark. And I think that if we have compounds that are important, then the solid state sensor world will make a sensor for it. I mean, they've done this for nitric oxide. They've done it for carbon monoxide. It's happened for methane. And I think if there's a need, the solid state sensor, I mean, I get calls almost weekly from solid state sensor people that are trying to develop sensors. And I think that that's the way to go for the future. And they're inexpensive. I mean, let's, almost throw away. So I think that is the way to go. I just don't think, but we need to know, first of all, what compounds are we looking for? What are diagnostic? And then we need to build sensors to measure them. That's what I believe in.
Yeah, I totally agree with that. Actually, when we first started working in the breath fields, so obviously we had a spectrometer-based sensor technology, and our assumption was that, what's being published, we can functionalize that already in the software to the chip. But then when you said, well, what is it? What's the biomarker? What's the panel of biomarkers for, pick a disease? And actually, that's where the big question mark was. So I think, once those things are known, I totally agree with you in the sense that there will be sensor-based solutions, whether it's spectrometry-based or pure target type sensors that will be able to step in and analyze those compounds. You just need to know what they are, basically. So we've got time for one more question, which kind of relates back to the points that you brought up here, which, obviously in adjacent fields like metabolomics, there have been a lot of learnings and best practices put in place there. What's the kind of top tips that the breath field should adapt, copy, or emulate from some of those adjacent fields? So maybe we could start with you on that one, Pierre.
Yeah, I think for me, like what we can really gain from the experience is the control of what we are doing. So we have to make sure that we get as many things as possible under control. So we discuss a lot about stuff that we cannot control, like microbiome. But as an analytical chemist, I have the feeling that something that we need to keep under control is the method. So we need to keep under control the sampling, the instrument, have robust QC. I know that we cannot generate a pool QC of breast, it's difficult. We don't have reference material for breast, but we have to be creative using some gaseous standard to monitor instrument stability, make sure that we don't have overtime batch effect and all these things that we know will happen, but they kind of are more difficult to track with VOCs. So I think if we keep control on the analytical process and the data processing side, we will improve a lot the robustness. And it's there that we can start to build something strong and progress in compound identification and sensor building and everything.
Excellent, thanks. Rianne, do you want to pick it up?
Yeah, my main idea behind this was actually QC, but Pierre already mentioned it. I think in metabolomics, they've really worked on quality control while measuring samples. I think this is being done more in breathomics now. When I did my PhD, not so much. So I think this is one of the main things we need to work on. And one of the things, I don't know, maybe this has been done in the past couple of years, but one thing that I also always had trouble with, if that's the right word to say, is that we don't really know how comparable different, for example, mass spectrometry methods are, right? Do we get similar results? If we use a different parameter in the mass spectrometer, do we get different results or do we get the same results? That might have changed, but that was something that bothered me back then, that even if you do, calculate or analyze the same samples with a different machine, totally different things pop up, which is a big issue, I think, because how are we going to standardize it if that happens?
Is that not the same in blood-based metabolomics? So again, if you're looking at,
have those studies where they try to optimize them and use multi-center studies, for example, with different machines and different labs to make sure that they find the same thing. And if you want to apply it clinically, you can't rely on one lab to analyze all the samples. It's just not feasible. So there needs to be some type of, I don't know, standardization might not be the right word, but some sort of commonality between different systems so that you can actually measure the same thing.
Mark, top tips from adjacent fields?
The only thing I guess I would add is I think setting a reporting standard is going to be quite critical going forward. I think it's still the case that the way many studies are reported make it difficult to assess what the value of the data is. And I think just building that framework, saying if you do a mass spec study, these are the things you report. In principle, you need to run standards. Otherwise, you need to be explicit that you haven't done so that these are tentative ideas, etc., to prevent the literature from polluting. And I think that will also help raise the standard. And that's certainly ongoing. But I definitely think there's a step we could make there. Most studies now have external validation, which is already a great step forward compared to the past, but it's definitely not the case for all studies.
Terence?
It's well known that depending upon whether you do negative ion, positive ion, which reaction gas you use in chemical ionization, and chemical ionization is just another name for SIFT and PTR mass spec, this makes a big difference in the signal. I mean, that's a fact of life. You could, if you are looking for molecules that are electronegative, using negative ions going to be much more sensitive. That's a fact of life and analytical chemistry. I mean, flame ionization detectors detect most sensitively hydrocarbons. This is known. This is not new science. This is old science. And it's a reality. If you pick an instrument that is selective for some molecules versus other molecules, that's what the data you're going to get to generate. So that's a function of the analytical chemistry. So I am not surprised. And it's the same if you pick whichever spectroscopy you use and whatever detector. So that's a fact of life. So I think that's real.
Excellent. Okay. Well, thank you so much, everyone. Really appreciate the time today. I'm looking forward to next year's panel when we can do it in person again. Fingers crossed for that vaccine. We can get back to more normal times and we'll be able to have a bit more interactivity with the audience. But thanks everyone as well for submitting questions.